Welcome back. Find a comfortable position and let the day begin to fall away. Tonight, let's slip quietly into the half-waking world of the sleepwalking mind. If these carry you off to sleep, subscribe. A new one comes every night. The body is already still. Breath has slowed. The chest rises and falls with the long, unhurried rhythm of something that no longer needs to be managed, something that has been trusted back to the machinery beneath thought. In the first minutes after sleep begins, the brain does not simply go dark. It reorganizes. It shifts. It begins a descent that is architectural in its precision, staged in its depth, and ancient in its logic, a logic that was already old before the first human closed their eyes against the dark. The descent happens in layers. Each layer has its own electrical signature, its own depth of quiet, its own particular relationship to the world outside. And the deepest of those layers, the one the brain is pressing toward in the first long hour of the night, is a place of such profound biological stillness that the word sleep barely contains it. In the brain's outermost sheet of tissue, the cortex, something is beginning to change. The high-frequency chatter of wakefulness, the fast, jagged electrical rhythms of attention and language and decision, is softening. The signals are slowing. They are growing wider apart, lower in frequency, larger in amplitude. What was a busy, irregular electrical landscape is becoming something calmer, something more like a tide. Sleep arrives in stages. The first is shallow, the barely there quality of early dozing, when a sound in the room can still pull the mind back to the surface in an instant. The limbs may twitch, the eyes move slowly beneath their lids. The brain is still near the surface, near the light. This first stage passes quickly, drifting down through a middle layer, where the brain becomes more seriously committed to sleep, where it begins to actively protect itself against the world outside, constructing small electrical shields against the interruptions that would otherwise pull it back up. And then, somewhere between 30 and 45 minutes into the night, the brain crosses a threshold into the deepest state it will reach. The electrical waves that move across the cortex become enormous. They arrive slowly at less than 2 full cycles every second, sweeping through the neural tissue the way long ocean swells cross open water, vast and unhurried and complete. This is slow-wave sleep. This is the deepest chamber of the night. The waves are called delta waves, and the name is almost too simple for what they represent. At less than 2 cycles per second, they are the slowest brain rhythms present in the sleeping mind, far slower than the rapid oscillations of wakefulness, slower even than the rhythms of the lighter sleep stages that precede them. Each wave is the coordinated firing of millions of cortical neurons oscillating together in a slow, massive pulse. Nothing in the waking brain looks like this. Nothing in lighter sleep looks like this. The delta wave is the signature of a brain that has fully surrendered, not to unconsciousness, but to a different and more fundamental kind of organization. In that first hour of the night, the slow waves come in abundance. They dominate the recording. They fill the trace. And though the body lying in the bed shows no sign of the electrical grandeur unfolding inside the skull, the face relaxed, the hands open, the breath moving in its long, slow arc, the brain is doing something of enormous biological consequence, organized around a precision that has taken a lifetime to calibrate. Sleep does not simply arrive. It is earned across every hour of the day by the slow accumulation of a single molecule. That molecule is adenosine. It is a byproduct of neural activity, a chemical trace left behind each time a neuron fires, each time the brain does the work of being awake. During waking hours, Adenosine gathers in the basal forebrain, the region at the brain's base that plays a central role in managing the transition between sleeping and waking. It accumulates gradually, hour by hour, in proportion to how long the brain has been running. A few hours of wakefulness produces a mild adenosine load. Many hours of wakefulness produces a heavy one, and that accumulating load is felt, not consciously, but in the deepening pull towards sleep that mounts across the day, in the heaviness that settles behind the eyes late in the afternoon, in the quality of the tiredness that arrives after a long and demanding day. A sleep scientist named Alexander Borbély gave this accumulating pressure a formal name and a formal model. In 1982, he described sleep regulation as a two-process system. One process is circadian, the roughly 24-hour clock governed by light and anchored in the brain's suprachiasmatic nucleus, which determines when the body is inclined to sleep and when it is inclined to wake. The other process is homeostatic. It is the adenosine load, the slow rising pressure of chemical sleepiness that builds from the first moment of waking and does not begin to clear until sleep begins. Borbély's insight was that these two processes work together, the circadian clock timing the window, and the homeostatic pressure determining how deeply the brain falls when the window opens. When the brain finally descends into slow-wave sleep, adenosine begins to clear. The pressure that has been building all day is finally allowed to resolve. In the depth and intensity of that first slow-wave sleep, the amplitude of those enormous delta waves, the completeness of the brain's commitment to this deepest stage, is directly proportional to how much adenosine has accumulated. A day of intense activity, sustained attention, and long wakefulness produces a large adenosine load. A large adenosine load produces a deeper, denser, more electrically intense first wave of slow-wave sleep. The brain's accounting is exacting. What was taken from it across the day is returned, with precision, in the dark. There is something almost architectural about the structure of a single night. The night has a shape. It is not a uniform descent into sleep and a uniform return to waking. It is a series of cycles, each lasting roughly 90 to 110 minutes, each moving through its own sequence of stages before returning, briefly toward the surface. Through the first cycle, the brain drops through the lighter stages and into slow-wave sleep, the deepest stage, the one defined by those vast slow delta waves. Then it climbs back up. Then it passes through a period of dreaming sleep before beginning the descent again. What shifts across the night is the proportion. In the first third of the night, slow-wave sleep dominates. The brain spends more time in the deepest stages, producing the highest amplitude delta waves, doing the most intensive biological work of the sleep period. By the second half of the night, slow-wave sleep recedes. The cycles become shorter in their delta content and longer in their dreaming content. The brain migrates from depth toward a different kind of activity, still asleep, but differently organized. The deep chamber left behind as the night moves on. This means that the hour or two after falling asleep represents something specific. Not just early sleep, not just the beginning of a process that will unfold more or less evenly across the night. That first descent into slow-wave sleep is the night's most critical biological event, the moment of greatest depth, the moment of greatest arousal threshold, the moment when more adenosine is clearing and more restoration is occurring than at any other point in the night. What happens in that first long wave of deep sleep is in some sense the point of the whole night. To reach the sleeper during slow-wave sleep requires more than a quiet sound. The arousal threshold in N3, the scientific designation for slow-wave sleep, shorthand for the third stage of non-rapid eye movement sleep, is the highest of the entire night. A noise that would wake a person in 5 seconds from lighter sleep might fail entirely to rouse them from slow-wave sleep. The brain, deep in its delta oscillations, is protected from the outside world in a way it is not at any other stage. That protection is not accidental. It is constructed. In the stage just before slow-wave sleep, the brain deploys two kinds of electrical event that serve as gatekeepers between the outside world and the deepening cortex. The first are sleep spindles, brief rhythmic bursts of electrical activity that appear on an EEG trace as tight, fast whorls, each one lasting a second or two, flaring out of the background rhythm and then fading back into it. A spindle looks on the recording like a brief bright signature, like a small flame, and like a flame, each one does something. In this case, it interrupts the passage of sensory signals from the thalamus to the cortex, actively dampening the brain's own responsiveness to the outside world. The thalamus is the brain's central relay, a paired structure deep in the center of the skull that receives incoming signals from the senses and decides in ordinary waking life which ones are worth passing upward to the cortex. During sleep, the thalamus shifts its role. Instead of relaying signals upward, it begins to generate rhythms of its own, rhythms that during the lighter non-REM stages produce those characteristic spindles and that help insulate the sleeping cortex from the stream of environmental information that would otherwise reach it. The spindle is the thalamus doing its gatekeeping work. Each one seals the brain a little more completely against the outside world. Alongside spindles, the brain in lighter non-REM sleep generates a second kind of electrical event, large distinctive waves called K-complexes. Where a spindle is rhythmic and repeating, a K-complex is singular and sharp, a large negative then positive deflection in the EEG trace that arrives sometimes spontaneously and sometimes in response to an external noise. The K-complex is thought to serve a dual function, both detecting a potentially significant stimulus and then suppressing the full arousal that might otherwise follow. It is the brain registering a sound in the hallway and then deciding, electrically, not to wake. Together, sleep spindles and K-complexes make N2 sleep, the stage that precedes slow-wave sleep, a carefully managed transition. The brain is not simply passive in this stage. It is actively working to protect the depth it is about to reach. The spindles fire and fade. The K-complexes register and suppress, and underneath, the delta waves begin to appear, first intermittently, then with increasing frequency and amplitude until they dominate the recording entirely, and the brain is fully in slow-wave sleep. The gatekeeping stages have done their work. The deepest chamber is now occupied. In the body below the brain, the descent into slow-wave sleep produces its own quiet changes. Core body temperature falls by nearly a degree across the first hour of sleep, a drop that reflects the body's withdrawal from its metabolic activity, the redistribution of blood flow toward the skin to release heat, the whole organism beginning to run its systems at a lower and less energetically costly rate. Heart rate reaches its nightly minimum, breathing becomes slow and regular, muscle tone decreases. The body is not simply resting, it is reducing, pulling back from the levels of activity that sustain wakefulness and settling into a metabolic quiet that is the necessary counterpart of the brain's deepest electrical state. In this metabolic quiet, one of the body's most significant hormonal events of the entire day takes place. The anterior pituitary gland, a small structure at the base of the brain, no larger than a pea, connected to the hypothalamus above it by a slender stalk, releases growth hormone into the bloodstream in its largest pulse of the night and of the day. The timing is not coincidental. The release of growth hormone during slow-wave sleep is tightly coupled to the depth of the delta oscillations above. The deeper and more complete the slow-wave sleep, the more substantial the hormonal pulse, and the effects of that pulse spreading through the body via the bloodstream in the hours of early sleep are not confined to the growth of children. In adults, growth hormone drives the repair of muscle and connective tissue, supports the maintenance of bone density, governs the metabolism of fats and glucose, and contributes to the immune system's nighttime work. It is the body's most concentrated investment in its own repair, and it arrives in the dark during the deepest electrical quiet of the brain as the delta waves move their slow and massive rounds through the cortex. None of this is visible. The body lying in the bed shows nothing of it. The face is still, the hands are open and loose, the breath is slow and untroubled. Everything that is happening, the enormous delta oscillations sweeping through the cortex, the thalamic gating protecting the depth from the outside world, the growth hormone moving through the blood, the adenosine gradually releasing its accumulated pressure into the clearing, all of it is hidden. It belongs to the inside of the night. It is the most biologically consequential and least observable passage of the day. The architecture of the first 90 minutes of sleep, the descent through lighter stages into the deep chamber, the appearance of the delta waves, the hormonal pulse, the protected quiet, has been shaped by a logic far older than any individual body. Sleep is not a modern invention. Its deep structure, the alternation of stages, the concentration of slow-wave sleep in the early night, the homeostatic pressure that calibrates its depth, is present across mammals in forms that suggest it evolved long before human brains existed. Whatever slow-wave sleep is doing, it has been doing it for a very long time. The mechanisms have been refined and preserved across millions of years because what they accomplish is necessary, not convenient, not beneficial, necessary in the way that eating is necessary, in the way that breathing is necessary. What they accomplish is not fully settled. The restoration of neural circuits, fatigued by the day's firing, the consolidation of what was learned while awake, the clearance of metabolic waste from the brain's extracellular space, the resetting of synaptic strength across the cortex so that the brain does not simply accumulate stronger and stronger connections indefinitely, but is periodically returned toward a state from which new learning can occur. These are the leading ideas, each supported by real evidence, none complete on its own, and the full accounting of what slow-wave sleep does for the brain remains one of the genuinely open questions in neuroscience. What is not open is the fact of the need. Deprive a brain of slow-wave sleep, and the methods for doing so can be precise. A sound at the right moment, timed to the appearance of delta waves on a monitoring device, can prevent entry into the deepest stage without fully waking the sleeper, and the consequences accumulate quickly. Reaction time degrades, memory consolidation falters, the capacity for sustained attention narrows, and the brain, given the chance to recover, will press harder into slow-wave sleep on the following night, producing deeper and more abundant delta oscillations, working to recover what was lost. The homeostatic pressure that Borbély described works in both directions. What is taken is sought back. The adenosine that has been building since the first moment of waking is at this point in the night releasing. It is being cleared from the basal forebrain at a rate that depends on the depth and completeness of the slow-wave sleep. The longer and more complete the delta oscillations, the faster the clearance. The brain is not simply running down and then being recharged in the morning. It is actively resolving a chemical imbalance that has accumulated through the work of the day, calibrating its own recovery with a precision that no external instrument could match. This is what Borbelli's two-process model describes in its formal terms. But stripped of its formality, it describes something simpler and more ancient. The brain runs. Running costs something. The cost accumulates in a single molecule, slowly across all the hours of the day until the pressure is sufficient to close the eyes and begin the descent. And then, in the deepest electrical quiet of the night, the cost is paid. The molecule clears, the pressure resolves, and the brain that emerges from the first long wave of slow wave sleep is not identical to the one that entered it. It is, in some specific and measurable sense, restored, not to the condition of an earlier age, not to any ideal, but to a closer approximation of its own functional baseline, the state from which the next day can begin. There is a precision in this that is worth pausing over, not the dramatic precision of a design system, not the precision of intention. The brain does not intend any of it, no more than a river intends its course, but the precision of something that has been selected iteratively over an enormous span of time for a result. The adenosine load calibrating the depth of the next night's slow wave sleep, the depth of the slow wave sleep calibrating the hormonal pulse, the hormonal pulse matching the tissue needs of the body, each element responsive to the others, each part of a system that has been tuning itself through variation and selection across generations toward a quiet and invisible competence. Outside the body, in the room, in the house, in the city beyond, the world continues. Traffic on distant roads, the small sounds of a building settling in the cool, the sky beginning, very slowly, to complete another rotation. None of it reaches the brain now. The thalamic gates are holding, the delta waves are slow and vast and complete. The growth hormone has entered the blood. The adenosine is clearing, and the deep chamber of the night is doing what it has always done, what it will do every night. The body is alive to enter it, paying the accumulated debt of the day back into the tissues and circuits and molecules that made the day possible. This is the architecture of the night's deepest passage, not dramatic, not visible, unhurried and patient and enormously precise, built from a logic that does not need to be understood to operate, that was running long before there were words for delta waves or adenosine or homeostatic pressure. It runs tonight the way it ran last night and the night before, and every night in every human life that has ever crossed through the dark and arrived, restored in some quiet fraction at the morning. The pressure that built all day is releasing now. Molecule by molecule, the chemical record of wakefulness is being settled. The brain is accounting for itself in the only way it can, in the dark, in the deep, in the slow electrical language of the delta wave, patient and enormous and wholly complete. In the image rendered by a research team in the year 2000, a human brain glows in false color against a dark field. The motor regions burn with warm light, amber and red, regions of active circulation, structures managing the body's movement through space. And at the front of that same brain, the prefrontal cortex sits in cool silence. Not dim, not reduced. Dark in the way that deep water is dark. Not empty, but unreachable, sealed away from the surface world, resting in a state that is unmistakably sleep. What makes this image worth sitting with, quietly for a long time, is that both of these conditions exist in the same skull at the same moment. The person this brain belongs to is walking. The researchers who produced that image, Claudio Bassetti and his colleagues, had captured something that sleep science had long suspected but never seen with such clarity. A sleepwalking brain is not a sleeping brain that has gone wrong. It is not a waking brain that has failed to rouse. It is something in between those two states, something the nervous system is capable of producing that has no ordinary name, because ordinary experience has no category for it. The motor systems are running, the body moves, the eyes are open, and yet the part of the brain responsible for knowing any of this is happening, for forming a memory of it, for judging whether it should happen at all, remains as deeply asleep as it was an hour before, when the body was still. The prefrontal cortex is a broad region of neural tissue sitting just behind the forehead. Its work during waking hours is continuous and largely invisible, the quiet background labor of monitoring behavior, suppressing impulses that should not be acted on, holding information in mind while a decision is being made, tracking the self across time, and comparing what is happening now against what has happened before. When it operates normally during wakefulness, its activity is so constant that its absence is almost unimaginable. And yet during a sleepwalking episode, that absence is precisely what the neuroimaging record shows. The blood flow has not been redirected there. The activity has not been sustained. From a functional standpoint, the prefrontal cortex has stayed in bed, even as the rest of the body has left the room. The consequence of that absence is not subtle. Without the prefrontal cortex engaged, there is no working memory, no ability to hold the thread of a situation in mind, to compare where one is now with where one was a moment ago, to construct a goal and pursue it through more than a few automatic steps. Without it, there is no inhibitory control, no capacity to evaluate an action and decide not to take it, no internal governor between an impulse and its execution. And without it, there is no self-monitoring, no part of the system looking at what the system is doing, registering whether it is appropriate, and redirecting it if it is not. The sleepwalker, who cannot respond meaningfully to a spoken question, who cannot remember in the morning what happened in the night, who moves through a familiar room without appearing to understand what they are doing there. All of that is the behavioral signature of a prefrontal cortex that is not present. The motor cortex, by contrast, is present, fully. Its circulation is maintained, its connectivity to the structures beneath it, the basal ganglia, the cerebellum, the brainstem pathways that drive coordinated movement, is active and capable of supporting complex behavior. The body can walk because the systems that organize walking are online. It can open doors because the motor programs for reaching and grasping are intact. It can navigate around furniture because the cerebellum, deep at the base of the brain, is quietly processing spatial information and calibrating balance and movement with its usual precision. None of this requires the prefrontal cortex to be awake. These systems are older, in evolutionary terms, than the cortex that sits above them, and they have their own competence, their own internal organization, their own capacity to manage the body's movement through a physical world. The cerebellum is a structure worth pausing on. Folded and dense, tucked at the back and base of the brain, beneath the great hemispheres of the cortex, it contains more neurons than the rest of the brain combined, and it manages the body's movement with a speed and a precision that conscious thought could never match. In ordinary wakefulness, its work is invisible, precisely because it succeeds. The hand reaches for a glass and finds it. The foot meets the floor at the right angle. The body sways and recovers without a moment of deliberate attention. During a sleepwalking episode, that work continues. The sleeping person who walks down a hallway and navigates around a chair in the dark is not doing something that requires cortical awareness. The cerebellum knows where the body is in space. It is calculating the adjustments needed to keep the body upright and moving. It does not need to wake the rest of the brain to do this. What the Bassetti neuroimaging study showed then was not simply a curiosity about sleep. It was a picture of something the brain can do that had not been clearly mapped before. Regional deactivation during sleep is not uniform. The brain does not go dark all at once, the way a house goes dark when the main switch is thrown. Different structures descend into sleep at different rates, in different sequences, and in the brains of people predisposed to sleepwalking, those structures can sometimes be running at different levels simultaneously. One region sleeps, another wakes partially. The body moves, the mind does not. The part of this picture that requires the most attention is what happens in the thalamus. During ordinary, undisturbed NREM sleep, the thalamus acts as the brain's gatekeeper. Sitting at roughly the center of the brain, it receives sensory information from almost every part of the body and the outside world, and serves as the relay station through which that information must pass before it can reach the cortex above. In normal slow-wave sleep, the thalamus does not simply receive and forward, it turns inward. Its circuits begin to generate a rhythmic, self-sustaining oscillation, the same oscillation that appears on an EEG as a sleep spindle, a brief burst of tight rhythmic activity that lasts for 1 or 2 seconds, flares across the recording trace, and then subsides. That spindle is the physical expression of the thalamus, dampening its own relay function. Sensory signals arrive. They are intercepted. The cortex is protected from disturbance. The brain goes on sleeping, even as the room outside continues to make sounds, even as light changes, even as the temperature shifts. During a sleepwalking episode, that gating mechanism appears to fail. Not completely. The cortex is still receiving some inhibitory protection, which is why the prefrontal regions remain asleep, but selectively, unevenly, in a way that allows certain signals to propagate upward through the thalamocortical system, while others are kept down. Motor signals move through. Arousal signals, the kind generated by the homeostatic pressure to surface from deep NREM sleep, find pathways upward, but the information that would activate the cortex's higher processing, the information that would light up the prefrontal regions and produce awareness and judgment and memory, does not complete the circuit. The gate is partly open and partly closed, and the result is a brain that is simultaneously more active than sleeping and less active than waking in different regions at the same time. The electrical record of that state is unlike anything produced by an ordinary sleeping brain or an ordinary waking one. When EEG electrodes are recording from a sleepwalker during an episode, the trace does not look like deep NREM. The slow, vast delta waves are still there, high amplitude oscillations rolling at less than 2 cycles per second across the recording, but moving alongside them in the same epoch are faster rhythms. Alpha frequencies, theta frequencies, the kind of activity normally associated with drowsiness, with light sleep, with the threshold states between one condition and another. These rhythms do not alternate, they coexist. The EEG trace during a sleepwalking episode carries both patterns at once, and the visual result is a kind of electrical layering, 2 distinct registers of brain activity, written across the same line of recorded time, neither one fully suppressing the other. This layering has a name in sleep research. It is called a dissociated state, a term that here does not mean anything psychological, but rather something strictly electrophysiological. Different regions and circuits of the brain are in genuinely different functional states at the same moment. The cortex maintains its slow NREM oscillations in some areas, Other circuits show the faster rhythms of partial wakefulness, and the thalamocortical system, the relay between them, is somewhere between its sleeping configuration and its waking one, generating the peculiar selective connectivity that allows the body to move while the mind remains absent. What researchers noticed, studying sleepwalkers across many nights in the polysomnography laboratory, was something that deepened this picture considerably. On nights when no behavioral episode occurred, when the sleepwalkers slept through from bedtime to morning without leaving the bed, the EEG still did not look entirely ordinary. The slow-wave sleep of sleepwalkers shows a higher rate of spontaneous partial arousals from N3 than the sleep of non-sleepwalkers. And interspersed within the deep NREM architecture of habitual sleepwalkers are what researchers have described as hypersynchronous delta bursts, surges of unusually high amplitude, high voltage delta activity that are not typical of standard slow-wave sleep and that appear to precede, or at minimum accompany, the kind of NREM instability that sometimes produces behavioral events. The brain that sleepwalks, in other words, carries a slightly different NREM architecture even when it is not sleepwalking. The instability is not intermittent. It is chronic, low-level, present across the whole night, even on quiet ones. Antonio Zadra and colleagues, working across many years of video polysomnographic research, helped establish this picture in greater detail. Their recordings captured the electrophysiology of the moments just before and just during sleepwalking episodes with a precision that earlier tools could not provide. What the recordings showed was a consistent pattern, a period of high amplitude hypersynchronous delta activity in the N3 stage, followed by the partial arousal event itself, the eyes opening, the body moving, the behavioral episode beginning, while the underlying EEG retained its NREM character in the cortical channels that mattered most. The episode did not erase the sleep. It occupied the sleep. It moved through it like a current moving through water without displacing the water entirely. The prefrontal cortex in those recordings stayed quiet throughout, through the partial arousal, through the locomotion, through whatever the person did in the minutes that followed. It remained functionally deactivated. And this is the detail that explains almost everything observable about sleepwalking behavior. The person who sleepwalks is not pretending to be asleep. They are not in an unusual psychological state. They are not responding to a dream that is a different phenomena, a different mechanism, a different stage of sleep entirely. They are in a state where the parts of the brain that produce awareness, memory, judgment, and coherent goal-directed behavior are genuinely offline, and the parts of the brain that produce movement and postural control and limited sensory processing are genuinely active. What the body does under those conditions is not random. It is organized by subcortical systems that have their own logic, their own motor programs, their own capacity to respond to the immediate physical environment without requiring any of that to rise into conscious experience. The ancient circuits that carry this load, the basal ganglia threading through the base of the forebrain, the cerebellum balancing and timing every movement, the brainstem nuclei that manage arousal and regulate the body's basic posture, were doing their work long before the prefrontal cortex existed in its current form. In the long history of vertebrate nervous systems, the machinery for moving a body through space evolved hundreds of millions of years before the elaborate cortical structures that allow a brain to know it is doing so. In a sleepwalking episode, those deep old circuits are the circuits that are awake. The newer, more recently acquired structures, the ones that generate self-awareness, that encode experience into narrative memory, that allow a mind to reflect on what it is doing, are the ones that have not come along. There is something worth slowing down for in that image. A person walking through a dark room, guided by systems that have been managing vertebrate movement since before there were rooms, or nights as we understand them, or brains capable of remembering either. The cerebellum making its small, rapid corrections to balance and trajectory. The basal ganglia sequencing the motor programs needed to reach for a door handle, to step over a threshold, to turn in the direction of a familiar space. All of it proceeding without the part of the brain that would, if it were awake, be making meaning of the experience, placing it in the story of a life, wondering what time it is, registering the strangeness of being upright in the dark at this hour. That part is asleep. The rest of the brain is not. The question of what the sleepwalking brain is actually experiencing, if experience is even the right word, opens onto a territory that current neuroscience cannot fully illuminate. The prefrontal cortex is not the only structure involved in generating conscious awareness, and its suppression during a sleepwalking episode does not necessarily mean that every thread of inner life is absent. Some researchers have noted that sleepwalkers occasionally produce intelligible words or phrases during episodes, not simply vocalizations, but fragments of language that suggest some low-level processing is occurring. In rare cases, people have reported partial fragmentary sensations upon waking that they associate with what happened during an episode, though full episodic memory is nearly always absent. What these fragments suggest is not clear. The honest answer is that the contents of a sleepwalking brain, whatever is or is not happening inside the circuits that remain partially active, are not accessible to the person living through the episode, and are not fully accessible to the science that has tried to observe it from the outside. What is clear, from the convergence of neuroimaging and electrophysiological research, is the structural fact: the brain is in two places at once, not metaphorically, not approximately. The blood flow measurements show it. The EEG traces show it. The behavioral record, a body moving with coordination and apparent purpose while remaining entirely inaccessible to memory, shows it from yet another angle. The sleeping brain has the ability to activate some of its systems while keeping others quiet, to allow movement while suppressing awareness, to sustain subcortical function while the cortex rests. Sleepwalking is what happens when that ability, which may serve some functional purpose during ordinary sleep transitions, becomes extended, sustained, and behaviorally expressed. The thalamocortical disconnection model, the idea that the thalamus's failure to fully gate the ascending arousal signals during an episode of non-REM instability, creates the specific combination of subcortical wakefulness and cortical sleep, has remained one of the most consistent frameworks for making sense of all these findings together. It does not explain every detail. Sleep science rarely offers explanations that explain every detail, and the nature of a state that the subject cannot report on and that can only be observed indirectly through imaging and electrical recording means that some things will remain uncertain for a long time. The model offers what the evidence supports: a picture of the thalamus caught between its sleeping configuration and its waking one, allowing enough signal to reach the motor and arousal systems to sustain behavioral activity, while not enough signal reaches the higher cortical regions to bring awareness, memory, or judgment back online. The thalamus itself is worth imagining clearly. It is not a single structure, but a collection of distinct nuclei, each one specialized, each one with its own connectivity to different cortical regions and different subcortical partners. The pulvinar nucleus handles certain aspects of visual processing and attention. The mediodorsal nucleus connects to the prefrontal cortex and is involved in working memory and executive function. The ventrolateral nucleus relays motor information from the cerebellum to the motor cortex. These nuclei do not all behave identically. They do not all enter or exit their sleep states in perfect synchrony, and it is in that asynchrony, in the possibility that some thalamic nuclei might remain more functionally connected to their cortical targets during an episode of NREM instability, while others stay in their sleeping configuration, that the anatomy of a dissociated state begins to become legible. The ventrolateral nucleus stays connected to the motor cortex, and the motor cortex stays connected to the limbs. The mediodorsal nucleus does not stay connected to the prefrontal regions in the same way, and so the prefrontal cortex stays dark. The body moves. The mind does not know. This is not a failure. It is worth being precise about that. The sleepwalking state is not the brain failing to do what it is supposed to do. It is the brain doing something that its architecture makes possible under specific conditions, conditions of NREM instability, of homeostatic pressure, of an internal arousal signal that is strong enough to activate certain circuits, but not strong enough to complete the transition to full wakefulness. The brain of a habitual sleepwalker is not damaged. Its slow-wave sleep architecture is different from the average, and that difference creates, on some nights and not others, the conditions for this third functional state. The architecture of that state, subcortical activation, cortical sleep, selective thalamocortical gating, is now documented well enough to be described with confidence. And the person on the other side of that state, lying in bed in the morning with no memory of the night's events, perhaps aware only that the room has changed slightly, or that a door that was closed is now open, is not aware of having been anywhere unusual. The brain returned to full sleep after the episode. The cortex continued its slow-wave oscillations. Whatever state the sleepwalking brain occupied, it closed without leaving a mark that the waking mind can find. The episode is over. The sleep continues, and the brain, having briefly inhabited a state that exists between sleeping and waking, between stillness and motion, between the ancient subcortical machinery and the quiet modern cortex above it, the brain settles back into its deep NREM rhythms, unremarkable and slow, as if the whole interval had not occurred. The posterior cingulate cortex, which the Bassetti study showed with elevated perfusion during sleepwalking episodes, sits at the inner surface of the brain, along the midline, a region involved in spatial processing and in what some researchers describe as aspects of self-referential awareness. That it should remain active during a sleepwalking episode, while the prefrontal cortex does not, is a detail the research raises without fully resolving. The posterior cingulate may be contributing something to the sleepwalker's navigation through space, a low-level, non-reflective spatial awareness that allows the body to orient itself in a familiar environment without requiring the frontal regions that would normally manage that task with full attention, or its elevated perfusion may be part of the partial arousal process itself, a circuit brought online by the ascending wake signal that has not been fully suppressed. The honest answer is that the significance of that finding is still being worked out, and it points toward the possibility that the dissociated state of sleepwalking is more anatomically complex than any simple two-part picture. Cortex sleeping, motor system waking, can fully capture. What the full picture holds, in its current state, is this: the sleepwalking brain is neither asleep nor awake in the ordinary senses of those words. It occupies a functional state defined by the coexistence of deep NREM cortical activity and subcortical motor activation, by the selective failure of thalamocortical gating, and by the persistence of electrophysiological signatures from both sleep and partial wakefulness within the same recording epoch. Its prefrontal cortex is functionally absent, producing the amnesia and behavioral disinhibition that characterize every episode. Its cerebellum and basal ganglia are present and functional, producing the coordination and spatial navigation that make the episodes appear, from the outside, almost purposeful. And the thalamus, that ancient relay station sitting in the middle of everything, managing the flow of information between the brain's deep structures and its cortical surface, is doing something between its two ordinary modes, generating a partial, selective connectivity that allows the body to move through the world while the mind that would recognize the world remains, for the duration of the episode, entirely absent. There is something restful in sitting with that image if it is approached without anxiety. The brain contains layers. Those layers were built at different times, in evolutionary terms, and they are capable of being in different states simultaneously, under conditions that arise naturally in certain kinds of non-REM sleep. The deepest, oldest circuits, the ones that balance a body and move it through space, can continue their work while the newest, most elaborate structures rest. The architecture that makes sleepwalking possible is the architecture of the brain itself, hierarchical, layered, only partially integrated at any given moment, capable of states that do not fit neatly into the categories that waking experience makes available. The false color image from the year 2000, the motor cortex warm and active, the prefrontal cortex dark and still, is a picture of that architecture, showing something it does not often show. It is not a picture of disorder, though it is sometimes produced in the context of clinical research on a condition that can occasionally carry risk. It is a picture of the brain's own layered complexity, briefly made visible by a state that is neither one thing nor another, the sleeping brain and the moving body. The dark prefrontal quiet and the lit motor current, both present, both real, and both, within a few minutes, resolving, the partial arousal completing or receding, the body returning to stillness, the EEG settling back into the long, unmarked rhythms of undisturbed, slow-wave sleep. The thalamocortical relay dims back toward its sleeping configuration. The motor signals that had found pathways upward through the partially open gate are gated again, or simply subside as the episode ends. The posterior cingulate and the cerebellum and the basal ganglia follow the cortex back into the quiet of deep non-REM, and the whole brain, having briefly occupied a state between two ordinary states, falls back into the one it had been in before, vast, slow, delta-dominated, the oscillations crossing neural tissue in long, unhurried waves, the body still, the room dark, the night continuing without needing anyone to observe it. This is the third state, not sleep, not wakefulness, but a narrow and temporary functional territory between them, produced when the brain's internal arousal signal is strong enough to activate some systems and not others, when the thalamic gate fails selectively rather than completely, when the ancient motor circuits wake into a room the thinking mind cannot see. It does not last long. It leaves no memory behind, and when it closes completely, and the sleep that surrounds it continues as if the interval were simply part of the night, unremarkable and deep, the brain resting in the way it has rested for as long as there have been brains, old, quiet, and far from anything that needs to be known. In families where sleepwalking runs deep, the numbers carry a particular weight. A child born to two parents who sleepwalk through the night carries a risk somewhere between 60 and 80% of developing the same condition across a lifetime, not a certainty, but something close to one. A child with one affected parent carries a lower probability, still many times the background rate seen in the general population, and a child with no affected parent at all faces odds closer to 2 or 3%, present but small, and shaped more by chance than by inheritance. The figures come from twin studies and family pedigree research accumulated over decades, and what they describe, quietly and precisely, is a biology that moves through generations. First-degree relatives of sleepwalkers, siblings, parents, children, show a prevalence rate roughly 10 times higher than the broader population. 10 times, not 2, not 3. The number points to something embedded in the genome, running beneath the surface of family life in a way that no family member may ever consciously recognize, because the episodes happen in darkness and are gone by morning. A grandmother who wandered her kitchen in the 1950s may never have connected her behavior to the child now sitting upright in bed. 3 generations later, hands moving over the sheets in slow, purposeless repetition. The connection runs deeper than memory. It runs in the chromosomes. What exactly is being inherited is not yet fully resolved. There are almost certainly multiple genes involved, each contributing a small share to the overall probability, in the way that most complex traits are shaped, not by a single decisive variant, but by a gathering of small influences that add and interact. The architecture of that inheritance is still being mapped, but one candidate has emerged from the genetic research with enough consistency to warrant attention: a specific variant within a gene cluster on the 6th chromosome. The 6th chromosome carries, among many other things, a large and functionally important set of genes known as the human leukocyte antigen complex. These genes are better known for their role in the immune system. They encode the proteins that sit on the surface of cells and help the immune system distinguish the body's own tissue from foreign material. They are the genes that determine tissue compatibility in organ transplantation. They are central to autoimmune conditions. They are, in most contexts, immune genes. Their connection to the depth and architecture of sleep was not anticipated and remains only partly understood. But a study published in the journal Sleep in 2015 found that adults with chronic sleepwalking disorder showed a significantly elevated frequency of a specific variant within this complex, a variant designated HLA-DQB 1*0501, compared to control subjects without the condition. The mechanism through which an immune gene variant might influence the stability of slow-wave sleep is not yet clear. The human leukocyte antigen proteins are expressed in brain tissue as well as in the periphery, and some immune signaling molecules are known to interact directly with sleep regulatory circuits in the hypothalamus and brainstem. The finding does not mean sleepwalking is an immune condition in the conventional sense. It means that the genetic architecture of non-REM sleep regulation is wider and more entangled with other biological systems than a clean mechanistic picture would suggest. The 6th chromosome, in this light, is not just an immune map. It is also, in part, a map of the night. The HLA complex sits on the chromosome. It influences, through mechanisms still being traced, the oscillatory quality of deep non-REM sleep in some of the people who carry its variants. And those people, sleeping in unremarkable beds in unremarkable houses, may find that their deepest sleep is slightly more prone to interruption, not waking, exactly, but not staying wholly in sleep either. Something in the system tips. Something in the long, slow delta rhythms of N3 fails, intermittently and unpredictably, to hold its own boundary. There is another part of the biological picture, and it lives not in the genome, but in the chemistry of the waking and sleeping brain. Deep in the brainstem, running in a slender column Through the central gray matter is a cluster of neurons whose primary output is serotonin. This structure is called the dorsal raphe nucleus, and its projections extend upward and outward throughout the brain into the prefrontal cortex, into the limbic system, into the thalamus and hypothalamus, in a broad, diffuse network that modulates arousal, mood, attention, and the transitions between behavioral states. During waking, the serotonergic output is steady and relatively high. As the brain descends through the stages of NREM sleep, the dorsal raphe quiets, its neurons slow their firing, the serotonin concentration in the regions it supplies begins to fall, and this gradual neurochemical withdrawal is part of the normal mechanism by which the brain releases itself from the alert, responsive condition of wakefulness and settles into the slower, more isolated state of deep sleep. In most brains, this quieting is smooth and complete enough that the transition to N3 is stable and maintained. The serotonergic systems dim, the prefrontal cortex follows them into functional inactivity, and the architecture of slow-wave sleep proceeds without interruption. But in brains predisposed to sleepwalking, the evidence suggests this quieting may be incomplete or poorly timed in ways that leave the arousal systems of the brain slightly more active during deep NREM than they should be, not fully awake, but not fully disengaged either. A residual tone in the system, a threshold slightly lower than it needs to be for N3 to hold cleanly through the night. This serotonergic hypothesis is not established fact. It is the best current picture built from a converging set of observations, and the most clinically direct of those observations comes from pharmacology. Medications that raise serotonin levels or disturb the timing of serotonergic suppression, selective serotonin reuptake inhibitors, serotonin, norepinephrine reuptake inhibitors, and various serotonin agonists have been documented across clinical reports and case series to trigger or worsen sleepwalking episodes in susceptible individuals. These drugs are widely prescribed for depression, anxiety, and other conditions. They do not cause sleepwalking in everyone who takes them, and in many people, they cause no sleep disruption at all. But in those with an underlying predisposition, the alteration in serotonergic tone appears to disturb the architecture of NREM sleep in ways that lower the threshold for partial arousal. A system already prone to instability at the boundary between deep sleep and waking becomes, with additional serotonergic modulation, more likely to cross that boundary without completing the crossing. The dorsal raphe nucleus is small. It is a slender, pale structure in the brainstem, tucked into the central core of the brain's oldest tissue. Its neurons are few in number relative to the vast populations of the cortex above. And yet, through its diffuse projections, thin filaments extending outward like the slow spread of something fluid, it exerts a modulating influence over an enormous range of brain function. The nightly quieting of those projections is part of the mechanism of sleep itself. Where that quieting is subtly imprecise, the night may not stay still. The dopaminergic system enters this picture from a different angle, and its involvement points to a different set of circuits. The nigrostriatal and mesolimbic pathways, the great dopamine roads running from the brainstem's substantia nigra and ventral tegmental area into the striatum and the frontal lobes, are associated primarily with movement, motivation, and reward. Their role in NREM sleep regulation is less direct than that of the serotonergic system and less well characterized. But clinical observations have established a connection that cannot be ignored. Dopamine agonist medications prescribed for restless leg syndrome and for Parkinson's disease have been linked in case reports and pharmacological records to the induction of complex nocturnal motor behaviors, including sleepwalking. The pattern suggests that dopaminergic activity, or its pharmacological amplification, can destabilize the motor inhibition that normally holds the body still during NREM sleep. The nigrostriatal pathway, which coordinates movement during waking, may under some conditions remain too active during deep sleep, pulling motor function toward expression at a time when it should be fully suppressed. The circuits involved in sleepwalking predisposition, then, are not a single thread. They are multiple systems: genetic, serotonergic, dopaminergic, thalamocortical, each contributing to the overall probability of NREM instability through different mechanisms and at different levels of the brain's organizational hierarchy. What they share is a common consequence: a slow-wave sleep that is less firmly bounded, less stably maintained, and more susceptible to the partial upward shift of arousal that produces the sleepwalking state. In the sleep laboratory, the EEG traces of people who sleepwalk show abnormalities even on nights when no episode occurs. The delta waves are there. They're large, slow oscillations moving across the record as they should, but alongside them, appearing with greater frequency than in control subjects, are bursts of synchronized high-amplitude delta activity that rise sharply and break before stabilizing, hypersynchronous delta bursts, the signature of a system trying to hold on to deep sleep while something in its regulation is pulling it upward. Spontaneous partial arousals happen more often. The boundary between N3 and lighter sleep is traversed more frequently in both directions across the night. The corridal multi-direction of TARC, or architecture, is not absent, but it is less stable than it should be. In a brain like this, the conditions for a sleepwalking episode are not confined to the moments when an episode occurs. They are the fabric of the night itself, chronic and continuous. Now consider that most children sleepwalk. Not all, but a significant fraction. Estimates place the prevalence somewhere between 15 and 40% across childhood, with rates highest in the years between 4 and 12. The figure dwarfs the adult prevalence, which sits between 1 and 4%, depending on the population studied and the threshold of the definition applied. Something about the developing nervous system makes it dramatically more prone to this state. The answer has two parts, and the first concerns sleep architecture. In early childhood, slow-wave sleep is extraordinarily abundant. The delta waves recorded from a child's sleeping brain are not merely deeper than an adult's. They are larger in amplitude, more densely packed across the night, and more dominant in the overall sleep structure. A child's brain generates the most powerful NREM oscillations of an entire lifetime during these years. The quantity of slow-wave sleep in a given night and the intensity of the delta activity within it are both at their lifetime maximum in childhood and decline progressively across adolescence and into adulthood, continuing to fall through middle and old age. More slow-wave sleep, more intense oscillations, a brain spending more of each night in the deepest and most physiologically demanding NREM state, and correspondingly, more raw material for the instability that produces partial arousal events. A child's brain is not simply a smaller adult brain spending more time in N3. It is a brain in a fundamentally different regulatory state, one shaped by the enormous metabolic demands of growth and neural development. The slow-wave sleep of childhood is not incidental to that development. It is central to it. The high-amplitude delta oscillations of N3 are the brain's most intensive restorative activity, and the developing nervous system needs them in abundance. The same biology that makes a child sleep so deep is also the biology that makes that sleep less stably contained. The second part of the answer concerns the prefrontal cortex. The prefrontal cortex is the last region of the brain to reach full functional maturity. Its development continues well beyond adolescence. The structural and functional optimization of its long-range connections to other brain regions, including the thalamus and the motor system, is not complete until the mid-20s. The myelin sheaths that insulate its axons and allow their signals to travel fast and cleanly are still being laid down throughout childhood and adolescence, still thickening and consolidating across the teenage years. A 10-year-old has a prefrontal cortex that is structurally present and functionally active, but not yet fully optimized. Its capacity for sustained, precise inhibitory control over behavior, over attention, over the body's motor systems is not yet what it will become. In the context of NREM sleep, this matters enormously. The prefrontal cortex exerts, among its many functions, a top-down regulatory influence over the thalamus and the arousal systems of the brainstem. During deep sleep, this influence shifts. The prefrontal cortex is among the first regions to fall into functional inactivity as NREM deepens, and this deactivation is part of the normal sleep maintenance process. But the quality of that regulatory relationship, how precisely the prefrontal cortex holds its boundaries with the arousal systems during sleep, may depend on how fully myelinated and optimized those long-range connections have become. In the child's brain, where the myelin is still incomplete and the connections are still being refined, the regulatory hold of the prefrontal cortex on the thalamocortical arousal system during N3 may be correspondingly less precise. Not broken, the child's sleep is, in most cases, functionally normal, but operating with less regulatory precision than the fully mature adult system. The combination is elegant in its logic, even if it does not feel elegant to the parents who check on a child found standing at the foot of the stairs in the middle of the night, eyes open, unresponsive, moving hands that have no task assigned to them. An immature prefrontal regulatory system, paired with the most abundant and intense slow-wave sleep the brain will ever generate, operating in a nervous system whose thalamocortical connections are not yet fully insulated. These are exactly the conditions that would be expected to produce more frequent, more easily triggered partial arousal events than a mature brain would generate under the same homeostatic pressures. And then gradually, the conditions change. The prefrontal cortex myelinates. Its long axons acquire their insulating sheaths one segment at a time, over years, in a process so slow that no single month shows measurable progress, and yet across a decade, the difference is complete. The regulatory connections to the thalamus become faster, more precise, more capable of holding the boundaries of NREM sleep cleanly against the ongoing pressure of the homeostatic and arousal systems. At the same time, the amplitude and abundance of slow-wave sleep is declining, the brain's homeostatic calibration adjusting across adolescence as the most intensive phase of neural development recedes. The delta waves of a 15-year-old are already smaller and less numerous than those of a 10-year-old. At 20, they are smaller still. The brain is, in this sense, growing itself into a different relationship with its own deepest sleep, gradually, across years, without any awareness on the part of the person doing the growing. For most children who sleepwalk, this maturation is the resolution. The episodes that were common at age 8 become rare at 12 and absent by 15 or 16. Not because anything was treated, and not because any deliberate intervention shaped the outcome, but because the brain completed the long developmental process that was always the underlying condition. The prefrontal cortex finished growing. The slow-wave sleep receded to its adult level. The thalamocortical system found its more stable configuration, and the night became quieter. This picture, childhood sleepwalking as an expression of normal developmental neurology rather than pathology, is one of the most reassuring findings in the sleep science literature. It places the prevalence of sleepwalking in children in its proper context, not as a sign that something has gone wrong, but as a consequence of the particular biology of a brain that is still, in its patient and unhurried way, completing itself. The twin who sleepwalks in one city, unaware that the other twin is doing the same thing on the same night in another city. This image carries the weight of what inheritance means in practice. Two nervous systems, built from the same genomic blueprint, running through the same NREM architecture in their separate darknesses, the same hypersynchronous delta bursts, rising and breaking in both EEG traces, the same thalamocortical boundary failing to hold at the same phase of the night, neither of them aware, across the distance, of what they share, neither of them aware, even in their own bedrooms, of what has happened. The inheritance is not in the memory. It is not accessible in daylight, in conversation, in any of the ordinary ways that family resemblance makes itself known. It is in the oscillatory pattern of the sleeping brain, expressed only in the dark, recorded only when someone is watching, a shared neurological signature running silently through a genome and expressing itself, night after night, in the deepest chamber of sleep. The serotonin-producing neurons in the brainstem that dim too slowly, the dopaminergic circuits that keep the motor system faintly available when it should be fully quiet, the HLA variant on the 6th chromosome influencing through mechanisms still being traced the oscillatory quality of N3, the delta waves of childhood arriving in amplitude so large they press the regulatory system past the boundary. It is not yet equipped to maintain precisely. Each of these is a piece of the same picture, seen from a different level of magnification: the molecular, the cellular, the circuit, the developmental. They do not all operate at once in every affected brain. They do not produce identical outcomes in everyone who carries them. The biology of predisposition is not a single mechanism, but a landscape, and the path through it varies. What they converge on, from all these different levels, is the same conclusion. Sleepwalking is not the brain failing to sleep. It is the brain sleeping in a particular way, shaped by a particular biological inheritance, at a particular moment in a long developmental arc. The episodes are an expression of that biology in the hours when no one, including the person sleeping, is watching. They are a consequence of deep NREM sleep being genuinely deep, so deep that the full architecture of waking consciousness is not available to contain or monitor what happens within it, and of the regulatory systems that normally hold that depth in place being, in some individuals and some moments, slightly less precise than the task requires. Somewhere a child is sleeping in the way children sleep, enormously, completely, the delta waves crossing the cortex in wide, slow arcs that dwarf the oscillations the same brain will generate 30 years from now. The prefrontal cortex is quiet, as it should be in N3, and its long-range connections to the thalamus are carrying signals through axons still in the process of acquiring their full insulation. Everything is functioning. Everything is developing on its own schedule, in the unhurried way that biological maturation always proceeds. The architecture of sleep is not broken. It is simply, in this phase of life, the architecture of a brain that has not yet grown all the way into the regulatory precision that the adult night will eventually require, and the brain is growing toward it, slowly, in the dark, in the tissue that never stops reorganizing itself across the long years of development, laying down myelin, refining connections, calibrating the thalamocortical system's sensitivity to the ongoing pressure of arousal, not with any urgency, not against any deadline that the brain itself recognizes, simply proceeding in the way that growing things proceed toward a configuration that will one day hold the boundaries of deep sleep more cleanly than they can be held right now. The dorsal right nucleus rests in the brainstem, its serotonin-producing neurons still and quiet in the depths of N3. The long axons of the prefrontal cortex reach downward toward the thalamus through pathways that are, tonight, a little more insulated than they were a year ago. The genome on the sixth chromosome carries whatever it carries, expressing it through mechanisms that remain partly open questions, shaping the oscillatory landscape of the night in ways that are not visible and not felt and not remembered. The delta waves come in slow and large, as they have every night since early childhood, filling the EEG record with their wide arcs, and the brain rests inside them, not perfectly, not with the stability it will one day achieve, but deeply, genuinely, in the only way it currently knows how. That, too, is a kind of becoming. The imprecision of the developing system is not a failure. It is the brain in an earlier state of itself, running the most ancient and necessary program in biology, the program of growth, on a time scale too slow to observe, but too fundamental to stop. The night will be what it will be, and the morning will come, as it always does, without memory of whatever the body found its way through in the hours before. The prefrontal cortex will keep growing, the axons will keep acquiring their sheaths. The thalamocortical system will find, year by year and in increments too small to name, a more stable configuration. The delta waves will slowly become what the adult brain produces, still large, still necessary, still the signature of the deepest and most restorative sleep the body is capable of, but a little less likely, as the years pass, to carry the regulatory system past the boundary of its own containment. And somewhere in that long, quiet, patient maturation, in the laying down of myelin, and the refinement of connection, and the slow calibration of circuits that will eventually hold the night more firmly in place, the episodes that were common will become infrequent, and the infrequent will become rare, and the rare will eventually not come at all. Not because anything ended, because something finished growing, on a timescale the brain keeps entirely to itself, in the dark that surrounds every sleeping body on every ordinary night. The body has been awake for 16 hours. Not dramatically awake, not unusually so, just the ordinary wakefulness of a day, with its small frictions and its long stretches of routine, its meals and its light and its sounds arriving and departing without ceremony. And in that time, quietly, invisibly, a pressure has been building, not in the muscles, not in the eyes, though those register it too, in the brain itself, in the deep forebrain structures that have been counting the hours since morning, noting each one with the slow accumulation of a single molecule, patient and precise, waiting for the moment the body finally lies down and the long surrender begins. That molecule is adenosine. It asks for nothing. It simply gathers. By the time the body finally settles into the first hours of the night, adenosine has built to a concentration that pulls the sleeping brain down into slow-wave sleep with unusual force. The descending pressure of a long day's wakefulness becomes the engine of an unusually deep night. And for most brains, on most nights, that depth is where restoration lives. The delta waves arrive at less than 2 cycles per second, vast and low and steady, and the body enters its most quiet state. Growth hormone moves through the bloodstream in the dark, core temperature falls, the heart slows, nothing disturbs the deep architecture of the night. For a brain that carries the particular vulnerabilities described across the earlier science of this subject, though, that depth carries something else alongside it. The same homeostatic force that drives the brain into slow-wave sleep also presses, in the predisposed, against the edge of instability. The deeper the sleep and the more forcefully it arrives, the greater the distance between that deep floor of non-REM and the partial arousal that may interrupt it. The greater the distance, the more disorienting that interruption when it comes. The precipitating conditions for sleepwalking are not dramatic things. Most of them are ordinary. Most of them accumulate the way adenosine does, slowly, quietly, and without announcement. The most direct route to that instability is the simplest one. A brain deprived of sleep carries more homeostatic pressure than usual into its first recovery night. Laboratory studies have confirmed what the logic of sleep biology already suggests, that subjecting an individual with an underlying predisposition to a period of sleep deprivation and then allowing recovery sleep reliably increases the frequency of sleepwalking episodes. The mechanism is not subtle. The brain's adenosine debt is larger than usual. The pressure driving it into slow-wave sleep is correspondingly greater. The delta oscillations of recovery slow-wave sleep are higher in amplitude, more consolidated, more intense, and the regulatory systems that normally contain that depth, the thalamocortical gating that maintains the boundary between non-REM sleep and partial arousal, are pressed against their limits from the beginning of the night. Think of it as a door the brain normally holds closed. On an ordinary night, with ordinary sleep pressure, the door holds without effort. The depth of slow-wave sleep is sufficient, but not extreme. The boundary between sleeping and that ambiguous middle state is clear. On a recovery night following sleep deprivation, the pressure behind the door is higher. The door holds for most brains, but in a brain where the regulatory architecture is already less stable than usual, where the delta oscillations are already prone to hypersynchronous bursts, where the thalamocortical system already fails more often than average at containing the transition between states, the pressure of recovery sleep becomes, for those hours, an additional risk. The recovery sleep does not cause the sleepwalking. It does not install any vulnerability that was not already present. It takes an existing predisposition and amplifies the conditions under which that predisposition expresses itself. The distinction matters. The brain that walks at night after a night of insufficient sleep is not a different brain from the one that sleeps through without incident. It is the same brain carrying the same architecture, encountering a circumstance that has raised the homeostatic stakes of its own deepest rest. Sleep deprivation comes in many forms. A single missed night, a week of shortened nights, each one ending a little too early or beginning a little too late, the debt accumulating the way adenosine does, without fanfare, without a clear threshold, quietly compressing the margin until the recovery night arrives and the pressure releases all at once. Travel across time zones, a shift change at work, a young child who does not sleep through. Any of these can create the conditions in which recovery slow-wave sleep is more intense, more consolidated, and less smoothly regulated than usual. For most people, the consequence of that intensity is simply a night of unusually deep, unusually restorative rest. For a brain already inclined toward non-REM instability, it is the circumstance most reliably associated with an episode. There is another route, physiologically distinct, but arriving at a similar destination. The body is ill, temperature has risen, not greatly, not dangerously, perhaps a degree and a half, the ordinary fever of an ordinary respiratory infection. The immune system is active, And part of what an active immune system does, part of the chemistry it releases into the bloodstream in the brain, is directly alter the architecture of sleep. Pro-inflammatory cytokines, molecules including interleukin-1 and tumor necrosis factor alpha, are not primarily molecules of sleep. They are molecules of immune signaling. Their purpose is to coordinate the body's defensive response to infection, but they also carry direct effects on the brain's sleep regulatory systems, binding to receptors in the hypothalamus and the basal forebrain, the very structures that govern the depth and character of slow-wave sleep. When those molecules arrive at those receptors in quantity, as they do during even a modest febrile illness, the result is a push toward high-amplitude delta sleep. The sleeping brain is driven down. The slow-wave oscillations are not merely present, they are intensified, sometimes dramatically so. The delta waves that move across the cortex during illness-related sleep are not the quiet, measured oscillations of ordinary deep NREM. They are larger, less evenly spaced. The architecture they create is physiologically intense but regulatorily unstable, driven by the immune system's chemistry rather than by the smooth progression of the brain's own homeostatic processes. For a predisposed brain, this chaotic amplification of slow-wave sleep is among the most reliable precipitants known. The association between fever and first-time sleepwalking episodes in children, a pattern well documented in the clinical literature, is not a coincidence of timing. It is a direct consequence of cytokine-driven delta instability, pressing a vulnerable NREM regulatory system toward partial arousal. The fever breaks, the immune system quiets, the cytokines withdraw from the brain's regulatory circuits, and in most cases the sleepwalking episodes that accompanied the illness resolve alongside the fever, the regulatory system returning to its baseline once the inflammatory chemistry no longer artificially amplifies the slow-wave oscillations. The fever was not a cause in any permanent sense. It was a condition, temporary, physiological, and entirely legible once the mechanism is understood. Not all of the conditions that push a predisposed brain toward partial arousal are as transient as fever or as straightforward as sleep deprivation. Some arrive slowly and persist. The body under sustained psychological stress is a different physiological environment from the body at rest, and the differences run directly through the architecture of sleep in ways that are not always obvious from the outside. The hypothalamic pituitary adrenal axis is the body's central stress response system. Under ordinary circumstances, it cycles with the day, cortisol rising toward morning, falling through the afternoon, reaching its lowest point in the early hours of sleep. Under sustained psychological stress, that cycle does not disappear, but it is altered. Cortisol levels remain elevated relative to baseline. The sympathetic nervous system maintains a slightly higher tone across the hours when it would ordinarily be most quiet, and the effects of that persistent low-level activation on sleep architecture are measurable, reproducible, and directly relevant to the conditions that favor sleepwalking. Elevated cortisol increases the proportion of light, NREM sleep, and reduces the restorative quality of slow-wave sleep. It increases the frequency of nocturnal arousals, brief, partial returns toward wakefulness that most sleepers never consciously experience, but that appear clearly on a polysomnographic recording as EEG disruptions, as shifts in muscle tone, as small accelerations of the heart rate. The net effect creates a paradox that the sleep medicine literature has documented carefully. The brain that has been awake under stress, carrying the heightened adenosine debt of a long, effortful day, arrives at sleep with strong homeostatic pressure toward deep NREM, and simultaneously, The cortisol and sympathetic tone it carries into the night pull it back from the depth it is physiologically driven toward. The brain is pressed at the same moment and through competing mechanisms both downward into slow wave sleep and upward toward lighter stages. For a brain without underlying NREM instability, this tension resolves without consequence. The regulatory systems manage both pressures, cycling through the night's architecture with somewhat less grace than usual, but without producing any behavioral episode. For a brain where that architecture is already less stable, where hypersynchronous delta bursts already appear more often than average, where partial arousals from N3 already occur with greater frequency, the competing pressures of heightened homeostatic drive and sustained physiological arousal create a chronically unsettled NREM environment. The thalamocortical boundary that must hold the brain in deep sleep against the pull of partial arousal is engaged on two fronts simultaneously. The probability of a failed transition rises with each night the stress continues and falls gradually without sharp edges as the sustained arousal itself slowly diminishes. There is something in that pattern worth sitting with for a moment. The brain is not indifferent to the conditions it inhabits. The conditions accumulate in it, the way the day accumulates in the body, not always visible, not always felt in the moment, but present in the quality and regularity of the night that follows. The stress that a person carries into sleep does not vanish at the boundary between waking and the first stage of NREM. It persists, altered in its expression, rerouted through physiological channels that register it in different terms: cortisol, sympathetic tone, arousal threshold, and those channels shape the night's architecture from within. There is a category of precipitating condition that arrives not from the body's own chemistry, but from substances introduced to it, medications taken precisely as prescribed, for reasons unconnected to sleep. The relationship between certain pharmacological agents and the stability of NREM sleep is not simple. A drug that produces sleep, as measured by standard clinical criteria, does not necessarily produce sleep of normal architecture, and the architecture of slow-wave sleep is precisely the domain where the difference becomes consequential. The class of medications known as Z drugs, including zolpidem, azopiclone, and zalplon, are among the most widely prescribed sleep aids in clinical use. They work by enhancing the activity of GABA A receptors in the brain, increasing the inhibitory tone of the cortex in a way that produces measurable reductions in sleep latency and increases in total sleep time. In those terms, they do what they are designed to do. The EEG of a person sleeping under their influence shows reduced activity in the frequencies associated with wakefulness, and the behavioral evidence that the person falls asleep and stays asleep aligns with the clinical intention. The EEG of that same person during what the recording labels as NREM sleep tells a more complicated story. The slow-wave oscillations present under Z-drug influence are not architecturally identical to the slow-wave oscillations of undrugged NREM sleep. The precise character of the delta rhythm, its regularity, its coupling with the other oscillatory events of deep sleep, its relationship to the thalamocortical regulatory dynamics that maintain the boundary between NREM and partial arousal, is altered in ways that do not fully appear in the summary statistics of sleep staging, but that matter to the regulatory systems that must navigate the night. In a brain already predisposed to NREM instability, this pharmacological distortion of slow-wave architecture can raise rather than lower the probability of a partial arousal event. The pharmacovigilance record accumulated over the years of widespread Z-drug use was sufficient, eventually, to prompt a formal regulatory response. The Food and Drug Administration issued its strongest warning category for zolpidem ezopicolone, and zaleplon, specifically citing complex sleep behaviors, sleepwalking among them as a serious documented risk. The warning was not hypothetical. It reflected a pattern of reported episodes, substantial enough to require the clearest possible signal to prescribers and patients that these medications, despite their apparent sleep-promoting effects, carry a real probability of destabilizing the very NRM architecture they appear to produce. The appearance of sleep on an EEG and the healthy, stable regulation of NRM slow-wave sleep are not the same thing, and in a predisposed brain, the difference between them is navigated each night without the sleeper being aware that anything unusual is occurring until the morning arrives and the evidence of an episode visible to a bed partner or left in the room's rearranged details provides the record that memory cannot. There is a distinct and important precipitating mechanism that operates not through pharmacology or immune chemistry or the psychophysiology of stress, but through the repeated interruption of breathing itself. Obstructive sleep apnea is a condition in which the upper airway collapses repeatedly during sleep, each collapse producing a partial or complete cessation of airflow that lasts from a few seconds to a minute or more before the sleeping brain generates sufficient arousal to restore muscle tone and reopen the airway. In mild cases, this pattern may repeat dozens of times across a night. In severe cases, it may repeat hundreds of times, each event ending with a brief, partial emergence from whatever sleep stage was underway at the moment of collapse. Those brief emergences are not awakenings in any conscious sense. The sleeper does not remember them. They do not typically produce the behavioral evidence of full waking: no rising from bed, no calling out, no reaching for water. They register as micro-arousals on a polysomnographic recording, a brief shift in EEG frequency, a transient increase in muscle tone, a momentary acceleration of the heart rate, and then a return into sleep. Each one is brief. Each one is resolved. And if it were a single isolated event, its consequences for NRM architecture would be negligible. But in a person with moderate or severe obstructive sleep apnea, those micro-arousals are not isolated. They arrive repeatedly across every hour of the night, each one pulling the thalamocortical regulatory system briefly toward wakefulness before releasing it back into sleep. The cumulative effect of that repeated disruption on NRM architecture is substantial. The boundary that the regulatory system must maintain between deep slow-wave sleep and partial arousal is engaged hundreds of times across a night of untreated apnea. The thalamocortical gating mechanism, the relay system in the center of the brain that must hold the cortex in sleep against the upward pressure of arousal signals, is not breached once or twice. It is engaged and released and engaged again, hour after hour, in a pattern that steadily erodes whatever stability the NRM regulatory system began the night with. For an individual with an underlying predisposition to NRM instability, that erosion is particularly consequential. The apneic microarousals do not need to be the sole cause of sleepwalking. They need only be sufficient, cumulatively, to tip a regulatory system already inclined toward partial arousals across the threshold into behavioral expression. And the clinical evidence suggests clearly that this is precisely what happens. Multiple studies examining the co-occurrence of sleepwalking and obstructive sleep apnea have found rates of comorbidity substantially higher than chance. Adults who develop sleepwalking for the first time in middle age, long past the developmental period when the condition is most common, often prove, on investigation, to have undiagnosed obstructive sleep apnea of moderate to severe degree. The most compelling evidence for the apneic mechanism comes from what happens when the apnea is treated. Continuous positive airway pressure, a therapy that splints the upper airway open with a steady stream of pressurized air through a mask worn during sleep, eliminates the apneic events. The microaerosols disappear from the polysomnographic recording. The thalamocortical gating system is no longer engaged hundreds of times across the night. And in study after study, in patient after patient, the resolution of the microaerosols is followed by the resolution of the sleepwalking episodes, sometimes partially, often completely. The picture that emerges from that finding is precise and quiet in its implications. The sleepwalking was not a primary disorder in those cases. It was the expression in behavior of a regulatory system that had been chronically destabilized by a separate physiological process operating in parallel. Remove the destabilizing process, allow the regulatory system to return to its baseline, and the partial arousals that produce the behavioral episodes cease. The brain was always capable of maintaining the boundary between deep NREM and wakefulness. It simply could not do so while simultaneously managing hundreds of apneic interruptions across every night of the year. There is a kind of clarity in that, if the mechanism is followed all the way through, the brain does not produce sleepwalking because it is broken. It produces sleepwalking because a specific set of conditions, operating on a specific underlying architecture, has temporarily exceeded the regulatory system's capacity for smooth management. The conditions are not random. They are physiological, describable, and in several important cases, modifiable. The interaction between precipitating conditions and underlying vulnerability is not additive in any simple sense. It is not the case that a certain number of risk factors accumulated in sufficient quantity will reliably produce an episode. Different precipitating conditions engage different mechanisms. Sleep deprivation and recovery sleep amplify homeostatic pressure. Febrile illness destabilizes slow-wave architecture through cytokine-driven amplification of delta oscillations. Sustained psychological stress creates a chronic tension between homeostatic drive and cortisol-mediated arousal. Pharmacological agents distort the character of NREM sleep without suppressing the appearance of it. Apneic microaerosols erode thalamocortical stability across the entire night through sheer repetition. Any one of these conditions in a sufficiently predisposed brain may be enough. In a brain less predisposed, several conditions in combination may still produce no episode. The relationship between precipitant and outcome is probabilistic, shaped by the underlying architecture in ways that cannot always be predicted in any individual case. What can be said is that the conditions are real and that they do not operate invisibly. The effects of sleep deprivation on slow wave intensity are measurable. The effects of febrile illness on delta amplitude are measurable. The effects of elevated cortisol on NREM architecture are measurable. The pharmacological distortion of slow wave oscillation quality is measurable. The microaerosols of untreated obstructive sleep apnea are measurable. They appear directly on the polysomnographic record, countable, time-stamped, each one a small visible perturbation in the rhythm of the night. The mechanisms are not hidden. They are legible to anyone who knows how to read the signals the sleeping brain produces, and they connect each one directly from a known condition to a known consequence through a chain of physiology that can be followed step by step. There is something restful in that legibility. The brain that walks in the night is not acting from nowhere. Its behavior emerges from conditions, physiological, pharmacological, circumstantial, that have their own logic, their own traceable causes. The partial arousal from deep NREM is not arbitrary. It is the result of a regulatory system that has been in some specific and identifiable way pushed toward a boundary it would not otherwise have reached. That does not make the episodes less remarkable, but it makes them less alien and less unknowable than they might seem in the moment of occurrence. The night continues. The body that initiated its recovery sleep following a long period of insufficient rest descends again into deep NREM, the slow wave oscillations moving through the cortex at their characteristic pace, vast and low and steady. The thalamocortical gating system holds what it can hold. The adenosine pressure that built across the waking day releases gradually into the depth of slow wave sleep, as it always does, patient and precise, following the logic the brain has used for as long as there have been brains to use it. If the conditions have eased, if the fever has passed, line of the apnoic events have been silenced by a steady stream of pressurized air through a mask, if the cortisol has returned, night by night, toward its quieter baseline, then the regulatory system operates within its normal range, and the boundary between deep NREM and partial arousal holds without effort, and the night proceeds without event. The slow wave oscillations reach their first trough of the night, deep in the first ultradian cycle, 2 hours or less after sleep onset. The brain is at its most remote from waking. The growth hormone released by the pituitary moves through the bloodstream in invisible quantities, attending to the body's repair. Core temperature sits at its nightly low. The heart moves at its slowest rate. Everything in the organism's physiology points in the same direction, downward, quieter, more still. The thalamocortical relay at the center of the brain maintains the cortex in its sleep state, gating sensory information away from consciousness, holding the boundary steady against whatever small perturbations the night contains. When that system works, and when the conditions that challenge it are absent or mild, the sleeper passes through the night without interruption. The first ultradian cycle gives way to the second gives way to the third. The slow wave sleep, concentrated in the early hours, gradually yields across the night to the longer and longer periods of REM that mark the later cycles, the delta oscillations fading as the homeostatic pressure they were created to serve is progressively discharged. By morning, the adenosine that accumulated across the previous day has been cleared. The slow wave oscillations of the night before have done their work. The body is warmer, the heart is beginning its slow acceleration toward waking rates, and the cortex is preparing, gradually and without urgency, for the first light of the day. That is the night working as the brain intends. The precipitating conditions, the missed nights, the fever, the persistent stress, the pharmacological distortion, the apneic interruptions are the circumstances that make that clean arc less available. Not permanently, not inevitably, but for the hours or the nights or the weeks in which they are present, they raise the probability that the thalamocortical boundary will fail at some point between sleep onset and morning, that the motor systems of the deep brain will receive an incomplete and uncontrolled arousal signal, and that the body will begin to move through the night before consciousness has arrived to guide or witness it. The conditions are transient, most of them. The fever resolves. The missed nights are repaid. The stress, however sustained it feels in the middle of it, changes with time, the physiological arousal that accompanied it gradually following the circumstances as they shift, the cortisol finding its lower baseline again, the sleep architecture regaining whatever stability it began with. The medications can be reconsidered. The airway can be splinted open with steady pressurized air. The microarousals can be made to cease, and when they cease, something settles. The thalamocortical gating system no longer required to manage hundreds of apneic interruptions or navigate the distorted slow wave architecture of a pharmacologically altered night, returns to the task it was organized for, holding the cortex in deep sleep, maintaining the boundary between NREM and partial arousal, allowing the delta oscillations to complete their slow and necessary work without interruption. The night quiets, the boundary holds, the depth of slow wave sleep, no longer a condition of instability, but of restoration, does what it has always been designed to do. The brain descends. The adenosine debt, whatever its size, is met with the depth of rest appropriate to it. The gating holds. The motor systems are quiet. The prefrontal cortex sleeps undisturbed. And somewhere in the lower oscillatory frequencies of the deep forebrain, in the very structures where adenosine accumulated across the day and where sleep pressure finally released into the night, the ancient arithmetic of wakefulness and rest completes another cycle. The conditions that were present are no longer present. The boundary that was challenged is no longer being challenged. The brain that was pressed toward its edge has returned, without ceremony, to the center of its own calm depth. The night continues around it slow and dark and sufficient, holding the sleeping body in the particular stillness that follows when the disrupting force is at last removed. A person sits upright in a dark bedroom, some hours before dawn. The eyes are open. The hands move slowly over the surface of the blanket, smoothing it, gathering it, releasing it, then beginning again. The room is still. The body is breathing with the long, unhurried cadence of deep sleep, and the eyes, though open, though oriented toward the far wall, where the curtains hold back the faint gray edge of the street, are not looking at anything that could be said to be there. This is the simplest form a sleepwalking episode takes. No walking, no navigation, only a person sitting in partial darkness, hands moving in a slow, repetitive gesture, with no apparent purpose, the gaze forward and unseeing, the body present, and the mind entirely elsewhere. Episodes of this kind are brief. They end on their own, without intervention, often without the sleeper changing position or fully returning to lie down. The hands slow, the eyes close. The breathing, which was never truly disrupted, continues its long measured rhythm into the resumed dark. By morning, the minutes will have closed without leaving any mark. Not all episodes are this contained. The spectrum of what this state can produce is wide, and the difference between its quieter expressions and its more complex ones is not a matter of how asleep the brain is. The underlying neurology is the same, but of which subcortical systems happen to become engaged as the partial arousal propagates. In some episodes, the sleeper stands. In some, they walk. In some, they navigate rooms, descend stairs, open doors, move through spaces they have moved through thousands of times by daylight, and do all of this with a rough competence that is, on its surface, entirely indistinguishable from ordinary behavior. The difference is internal. The difference is what is absent. What is absent is the prefrontal cortex, not the person, the executive layer that during waking hours monitors, evaluates, suppresses inappropriate actions, and keeps the self in continuous relationship with the world it moves through. During a sleepwalking episode, that layer remains in its sleep state. The body navigates. The brain's deeper structures manage balance and movement and the rough avoidance of obstacles. But the faculty that would say this is unusual, or I am cold, or I should go back to bed, that faculty is not available. The episode runs on older machinery, and older machinery runs differently. Researchers who have studied the behavioral spectrum of these episodes have drawn a practical distinction between their minor and major expressions. Minor episodes are the ones that resemble the bedroom scene above, limited in scope, repetitive in character, physically contained, carrying little risk of harm. A person sitting up in bed and moving their hands, a person murmuring a few syllables that do not resolve into words, a person lifting and replacing an object from the bedside table, or folding and unfolding a piece of cloth in the dark. These behaviors are brief, often lasting only a minute or two, and they tend to resolve without the sleeper leaving the bed or becoming more fully aroused. They are far more common than most people realize because they leave no trace and generate no memory. Major episodes are another thing. In a major episode, the sleeper rises and walks. They may leave the room. They may move through a house with a purposeful quality, not the stumbling, confused movement of someone woken abruptly from deep sleep, but something more fluid, more automatic, more at home in the space. The word purposeful is not quite accurate because purpose implies intention, and intention requires a mind that is present. What the sleepwalker exhibits is something closer to behavioral momentum. The motor systems have engaged. A pattern of movement has been initiated, and because the inhibitory oversight of the prefrontal cortex is offline, there is nothing to interrupt it, redirect it, or interrogate it. The pattern runs until it exhausts itself, or until some stimulus from the environment, a sudden loud sound, a change in surface underfoot, the resistance of a door, introduces a new input that the subcortical systems cannot integrate smoothly, and the episode shifts. The degree to which a sleepwalker responds to the environment during an episode is one of the more curious things about the state. They are not entirely unresponsive. They typically avoid large obstacles, furniture, walls, the edges of stairs, though not always, and falls and collisions are among the most common sources of injury during major episodes. They may pause at a doorway. They may turn in response to a sound, not because they heard it in the way a waking person hears, but because the auditory input reached some deeper level of processing that influenced the direction of movement without producing anything like awareness. If addressed directly in a calm voice, a sleepwalker may produce a brief verbal response, a short syllable, a fragment of a word, something that sounds almost like an answer without being one. These responses are not signs of waking. They are the output of language systems that can generate simple sounds without the prefrontal cortex online to compose them into meaning. This partial responsiveness is mediated by the systems that remain active during the episode. The cerebellum, deep and ancient at the back of the brain, continues its work. The continuous fine adjustment of posture and balance, the calibration of movement through space. The basal ganglia, those deep paired structures involved in initiating and sustaining motor programs, remain engaged. The sensory motor cortex, which organizes the physical execution of movement, is active enough to guide the body through familiar environments. These are the systems that can turn a sleepwalker around when the wall is reached. They do not require consciousness to function. They have been functioning since long before the part of the brain capable of consciousness had fully formed. The eyes, during all of this, are open. In a darkened room, they appear to be oriented toward whatever is in front of the sleepwalker, tracking the space, reading the environment with the same quiet competence as the feet below them. But the gaze lacks the quality that distinguishes waking sight from its imitation. Waking eyes move in small, rapid shifts, tiny, precise adjustments that keep the region of interest centered, that compare and update the visual field, that continuously confirm the relationship between the body and the space it occupies. The eyes of a sleepwalker do not move like this. They are oriented, but not scanning. They receive light. They do not assemble what they receive into perception in the full sense of that word, because the association cortices that would transform raw visual input into recognized forms and meaningful relationships remain in their sleep state. The room is there, its shapes are registered, but the registration stops before it becomes understanding. This is what the hippocampus cannot do in the hours of an episode. The hippocampus, the brain's primary structure for encoding new experience into lasting memory, remains in its sleep state throughout the episode. Whatever happens, however long the episode lasts, however far the sleepwalker moves, however much the body interacts with the world around it, none of it crosses the threshold into memory. The encoding mechanism is not active, the event will not be stored. And because it is not stored, it cannot be recalled. When morning arrives, the night before is simply absent, not blurred, not fragmentary, but genuinely blank, the way a period of anesthesia is blank, because the machinery for registration was not running. This amnesia is total in almost every case. It does not feel, on waking, like forgetting. It feels like the night passed without incident, because for the conscious mind, it did. There is no sensation of a gap, no residue of interrupted sleep, no intuition that something occurred. The episode leaves no subjective trace, because subjectivity was not present during it. The self, in the philosophical sense of a continuous experiencing awareness, was elsewhere, suspended in the deep, slow rhythms of non-REM sleep, while the body moved through the house and returned and lay back down and released, quietly, into the resumed dark. A person navigating a darkened hallway during an episode walks with a gait that is slower than waking, but not dramatically so. The steps are careful in their way. The hands may trail along the wall, not searching exactly, but maintaining contact with the surface, the way a body that has lost one form of orientation maintains another. At the end of the hallway, there is a closed door, and the sleepwalker may reach for the handle and pause, and in the pause there is nothing, no deliberation, no recognition of the door as a door leading somewhere, no decision about whether to open it. There is simply a pause, and then either the hand closes on the handle or it does not, depending on something in the balance of motor activation and environmental input that is entirely beneath the level of thought. This is the quality of subcortical navigation. It is not thoughtless in the way we mean when we say someone acted thoughtlessly. It is anterior to thought. It is the kind of spatial competence the brain developed long before it had the capacity to think about space. The deep motor systems that guide a sleepwalker through a familiar house are the same systems that guide any skilled habitual movement, the reach for a glass, the turn of a key, the path from front door to kitchen, taken so many times that the feet know it without consultation. During waking hours, the prefrontal cortex overlays this automatic competence with awareness and intention and judgment. During an episode, the overlay is absent, and the automatic competence runs alone. The video polysomnography recordings made during monitored sleepwalking episodes in sleep laboratories capture the episode from two vantage points simultaneously. One channel shows the behavior, the body on the bed, the moment of sitting up, the quality of the movement as the episode develops, the duration, the return to the resting position. The other channel shows the neurophysiology, the EEG trace running alongside the video, recording in real time the electrical state of the brain as the episode unfolds. In the EEG, the moment of partial arousal is visible as an abrupt shift in the trace, the slow, large waves of deep non-REM interrupted by a sudden intrusion of faster rhythms that do not fully resolve into wakefulness, that hover in the middle range between sleep and waking. that produce the behavioral episode and then gradually give way as the episode ends and the trace returns to its slow, deep pattern. These two records, the behavioral and the neurophysiological, exist as parallel accounts of the same event. Neither is more real than the other. The body moving through the room and the electrical patterns running through the cortex are aspects of a single thing occurring in a single moment, one of them visible from the outside and the other only legible as a trace on a graph. The episode, seen this way, is not a disruption of sleep. It is a feature of a particular kind of brain in a particular phase of sleep, under particular conditions, a feature that can be measured, characterized, and understood, even if it cannot quite be placed within the ordinary categories of either sleeping or waking. The range of behaviors that major episodes can involve is wide enough that clinicians who study sleep disorders find it important to distinguish between the different forms the state can take. Some episodes involve behaviors that appear, from the outside, to be purposeful in a domestic sense, going to a kitchen, opening a refrigerator, handling food. Some involve complex sequences of physical action that unfold over several minutes, the body completing one part of a behavioral chain and proceeding to the next, each step triggering the initiation of the one that follows in a pattern that may be familiar and routine from waking life, running without oversight, because the part of the brain that provides oversight is asleep. Some episodes involve leaving the sleeping area altogether, descending stairs, moving through multiple rooms, in rare cases approaching an exterior door or window. The higher risk expressions of the state are not frequent, but they are not negligible, and they are the reason that careful clinical evaluation matters when episodes become complex or recurrent. A clinician evaluating such episodes is not only assessing the sleepwalking itself, they are ruling out other conditions that can produce similar-looking nocturnal behaviors through entirely different mechanisms. Nocturnal frontal lobe epilepsy produces complex motor behaviors during sleep that can look remarkably like sleepwalking, but arise from seizure activity rather than partial arousal from NREM. Or EM sleep behavior disorder, a condition in which the normal motor suppression of REM sleep fails, produces enacted dream behavior that is phenomenologically distinct from somnambulism, but can be confused with it in clinical settings. The EEG and the polysomnography are what separate these, the electrical signature of the brain during the episode telling the story that the behavior alone cannot tell. A sleepwalker who has been documented in a sleep laboratory wakes in the morning in the same condition as any other sleeper in the same laboratory. The night felt unremarkable. There may be a slight sense of not having slept as well as usual, or there may be nothing at all, no sensation of disturbance, no awareness of having been observed, no access to the episode that was captured in the video and the EEG trace. Asked about the night, they will say they slept. Asked about specific events, they will have nothing to report. The record exists, the experience does not. This asymmetry between what can be measured and what was lived is one of the more thought-inducing aspects of a sleepwalking episode, not because it implies anything was taken from the sleeper, but because it shows, quietly and precisely, how much of what the brain does at night occurs entirely outside the reach of the self that will wake in the morning. There is a version of the episode that ends with the sleepwalker returning to bed. This is the most common conclusion to a locomotor episode. The subcortical systems, having run their pattern, begin to wind down. The activation that sustained the episode gradually attenuates. The body, which has been moving through the house on the momentum of a partial arousal, slows and then stops, and in the stopping, there is often a kind of gravitational pull back toward the resting position, the bed, the familiar surface, the physical anchor of the sleeping place. The sleepwalker returns. They lie down with the deliberateness of someone who is, in some vestigial way, following a pattern of behavior whose final step is this one, and then they are still. The body settles. The breathing, which may have quickened slightly during the episode, resumes its deep and unhurried pace. The EEG trace, which showed the intrusion of faster rhythms during the episode, slowly fills again with the long, high-amplitude waves of deep non-REM sleep. The episode is over. Sleep continues. This closing moment is worth dwelling on. It is not a return to consciousness, followed by a return to sleep. It is a continuous state of sleep in which a behavioral episode occurred and concluded, with the brain's deeper regulatory systems managing the whole arc, the arousal, the behavior, the resolution, without the involvement of waking awareness at any point. The self that will wake in the morning was not present for any of it. The person who navigated the hallway and returned and lay down was not, in the sense that matters for experience and memory, anyone at all, only a body guided by systems that have been coordinating movement and spatial behavior since long before there was a self to be aware of them. Sleep disorders researchers who have spent years studying these episodes in the laboratory describe a quality of watching that is difficult to characterize. The episode is observable. The behavior can be videotaped. The physiology can be recorded. The arousal can be timed and measured and compared to baseline. And yet there is something in the image of a person moving through a familiar space with open eyes and an absent mind that resists easy description. Not because it is alarming. In the laboratory context it is generally not, but because it places the viewer in the presence of a very clear illustration of how much of the brain's activity runs below the level of the awareness that normally seems to be in charge. The body knows its house. The body knows how to move through the dark. The body does not need the self to do any of it, and the sleepwalking episode proves this gently and repeatedly in the quiet hours before dawn. The amnesia that follows is not experienced as loss. It cannot be, because the self that would register loss was not present when the episode occurred. There is no gap in the felt continuity of the night. The night simply ends and the morning begins, and the distance between them contains nothing that the waking mind can access. This is not the amnesia of forgetting something known, the word that slips from reach, the name that will not come. It is the amnesia of a mechanism that was not engaged. The recording did not run. The absence is clean. For people who experience recurring major episodes, and for the people who share sleeping spaces with them, the picture is more complex. The sleepwalker has no memory, but a partner or sibling or parent who witnessed the episode does. They remember the open eyes and the mechanical quality of the movement, and the failure of their words to reach the person they were addressed to. They may feel concern, or a kind of unsettled recognition that the familiar person was somewhere else for a while, that the body moving through the room was operating on different terms. This gap between the amnesia of the sleepwalker and the memory of the witness is itself a kind of document of the episode's strangeness, a record kept in human experience where the EEG cannot reach. The serotonergic and dopaminergic systems that influence the stability of non-REM sleep through the night, the systems whose fluctuations during waking and sleep shape the conditions in which a partial arousal becomes more or less likely, are not things that most people think about during the day. They are running quietly in the background of every waking hour, and they continue through the night, their activity gradually suppressed as non-REM deepens, and then gradually recovering as the cycles progress. In a brain where this suppression is imperfect, or where the transition between the suppressed and the aroused state is less smooth than it should be. The conditions for a partial arousal, a brief, incomplete emergence from deep NREM that activates motor systems without restoring cortical awareness, are present more often and are more easily triggered by the circumstances of a given night. What changes the likelihood of an episode on any particular night has been described in terms of the conditions that push the brain into deeper and more unstable NREM sleep. The accumulated pressure of insufficient sleep in recent nights, the delta-promoting effects of a fever, the architectural disturbance created by certain medications, the repeated micro-arousals of an untreated breathing disorder. When those conditions ease, the episodes tend to ease too. The brain, responsive and plastic in the ways that matter, adjusts its sleep architecture when the pressures on it change. The relationship is not simple, and the adjustment is not immediate, but it is real. A brain that has been operating under conditions favorable to partial arousals will, when those conditions improve, generally produce fewer of them. The episodes are not a fixed feature of the person. They are a feature of the brain under particular conditions, and conditions change. Across the years of childhood, those conditions change in a specific way. The enormous, high-amplitude, slow-wave sleep of the immature brain, the deep NREM oscillations that make the child's sleeping EEG look so different from an adult's. The waves so large and so synchronized that they are among the most powerful electrical signals the brain will generate across an entire lifetime gradually attenuates as the brain matures. The prefrontal cortex completes its long myelination. The long-range connections between the frontal regions and the thalamus and the motor systems become better insulated and more precise in their timing. The regulatory architecture of NREM sleep stabilizes, and most children who sleepwalk regularly at age 6 or 8 or 10 find that the episodes become less frequent in their early teens and cease entirely before adulthood. The developmental window closes. The brain grows itself into greater stability, and the partial arousals that were a feature of the immature architecture gradually become rare and then absent. This is not universally true. In some people, the predisposition persists, shaped by genetics and by the particular architecture of their NREM regulation, and episodes continue into adult life, or begin for the first time in adulthood, usually in the context of specific precipitating conditions. But even in adults with chronic sleepwalking, the episodes are not continuous. They occur in clusters related to stress, illness, medications, disrupted sleep schedules, and other conditions that destabilize NRM architecture. Between those clusters, there are nights of undisturbed sleep in which the regulatory systems hold their balance, and the brain passes through its cycles from N1 to N2 to N3 to REM and back again without any partial arousal, any behavioral episode, any interruption to the long, slow, restorative business of deep sleep. On those undisturbed nights, the sleepwalking brain sleeps as any other brain sleeps. The delta waves spread across the cortex at less than 2 cycles per second, wide and unhurried. The motor systems are quiet. The thalamus gates sensory information away from the cortex, and the cortex accepts this gating and rests within it. The body moves through the cycles of the night, the temperature falling in the early hours, the growth hormone releasing in its first quiet tide, the adenosine pressure that accumulated across the waking day finally and fully dispersing into the depth of slow-wave sleep, and by morning the debt is paid and the balance restored, and the person wakes as if from nowhere, from nothing, from a darkness that holds no episodes and carries no residue. The chair in the corner of the bedroom where a sleepwalker might pause during an episode its shape registered by open eyes, its position integrated into the movement of the body navigating around it, stands in the same position during the undisturbed nights. The hallway beyond the door is the same hallway. The house is unchanged. And in the sleeping brain, the delta waves pass through the same tissue, the same neurons, cycling through the same slow oscillation. The deep systems maintaining the same ancient functions they have maintained through every night of the person's life. What differs on the nights without episodes is the stability of the transition at the top of N3, the cleanliness with which the regulatory systems hold the boundary between deep sleep and partial arousal, the absence of the conditions that would push them past that boundary. When that boundary holds, the episode does not occur. The body stays still. The eyes stay closed. The hippocampus encodes nothing because there is nothing to encode. The prefrontal cortex rests in its deactivated state, and there is no need for it to do otherwise. The whole brain moves together through the long descent into slow-wave sleep and back up through the lighter stages and into REM, and the night passes in the way most nights pass, unremarkably, invisibly, without event. The person who woke in a childhood bedroom to find themselves standing at the window, or who found the lights on in the kitchen the morning after a stressful week and understood what had happened, or who was told by someone who loves them that they sat up in the night and moved their hands over the blanket and said something that did not quite make sense. That person carries this history the way anyone carries the idiosyncrasies of their own nervous system, as a fact about themselves that arrived without being chosen, and that sits most of the time entirely quietly in the background of a life. The episodes are part of the brain's history. They are not the brain's whole story. A bedroom in the hours before dawn, the curtains holding back the early gray. A body on the bed, lying still, breathing with the long, slow rhythm of deep NRM sleep, the eyes closed, the hands at rest on the blanket,